Hi everyone, I am Saniya, and in this video, I'm going to show you how you can overlap genomic regions using the GRanges or GenomicRanges package on bioconductor in R. So if you have two regions of DNA and you want to find if there are any overlaps, how do you do that? So this is something that is super important as well. And I think that this will hopefully help you guys. So just a very quick review. So, we have our DNA, deoxyribonucleic acid. So DNA is stored in the nucleus of each cell. It's wound up and coiled in a butterfly-shaped chromosome. So we actually only see chromosomes in their distinct butterfly shape, like this before they divide as well, which is called metaphase. So, they duplicate the DNA and then the cells get ready to divide into identical daughter cells. And so this DNA is in each of our cells. We have 46 chromosomes, 23 pairs, one set from each of our parents, biological parents. So this is a very cute baby. So inside the cell, we have, these 23 pairs of chromosomes. And if you zoom in further, you'll see that, each chromosome consists of a single very long DNA molecule. And each gene on the chromosome is a particular segment of that DNA with instructions for forming the organism's characteristics. So DNA is made up of these nucleotides that are hooked together, A, T, G, and C. And the sequence is code for the formation of proteins, regulation, or for structural functions. So some we don't even know the function of. And what's really surprising is a lot of it, the DNA, we are still trying to figure out what is a function of the DNA. So not all of the genetic coding is for protein development. Some DNA coding affects regulation in the body or structural functions, and some coding has no function that we have found yet. So this is still an ongoing discovery, but this is sort of like at the DNA level, like just a revision about DNA. So what our main task is, again, so you can see over here, this is a person. So you see, this is a person here, and we can look at one of the cells, the trillions, around like 37 trillion cells in our human body, according to some recent estimates in the adult human body. So if we zoom in on a human cell, a eukaryotic cell, you can see mitochondria, the nucleus, endoplasmic reticulum, Golgi apparatus, there are even these tiny, tiny dots called ribosomes, which help take messenger RNA and make that into a polypeptide that can form a protein or part of a protein. So you can see if we zoom in the nucleus, we have this chromosome region here, and the chromosome is wound really tightly around, the DNA is wound really tightly around these histones. Wherever it's wound tightly around histones, we can't really access that DNA. We can't really transcribe it into protein. But in any case, so this is just DNA wound around, and you can see the bases, G pairs with C, guanine pairs with cytosine, and thymine pairs with adenine. So that's just what's happening along the DNA. So, one of the most common forms of human variation is a single nucleotide polymorphism. So that's called a single nucleotide polymorphism. And that's just one DNA base pair. It's just one DNA, one base pair. So in a specific location, in a specific DNA location, so this little smiley is a little bit puzzled. It's a SNP or it's a mutation. It's very common. So, and some people, this might be like in one population, they have this DNA, and the other population, they have this DNA, and in this location, like, a chromosome and a specific location. And in some people, it's an A at this spot, and other people, it's a G at this spot, so we look at one of the DNA strands and we see that, it's an A in this position, and in other people, it's a G at this position. And of course, the complementary base would be that this is a T here, for those people. And this would be a C here for those other people, the other DNA strand. So that would be the difference. This is a C. So we were trying to figure out, okay, well, what is this exactly, like what happens? So this is a T, that's a T, A, T, C, G, G by complementary, and the same thing here, but it's just that this is the difference. So the SNP has a location on a chromosome. And what happens is we want to figure out in this video, how can we figure out if a SNP, the SNP falls into an interval below. So imagine we have a DNA region, you know, we're looking along one of the chromosomes in our nucleus, just along one of the chromosomes, or it could be many different SNPs, but we're looking there and we're looking at one of these chromosomes along one of the DNA strands here. And we have this DNA here. It's a DNA region that we're looking at. It's a start and it's an end region. So we're trying to figure out, does this SNP at a specific location, does it fall into this interval below and within the region start and end? You know, can we draw, like, does it fall? Like, does it land right in the region or not? Does it land? Then if it lands, then yes. That's great. Otherwise, if it's falling like, if it's instead falling somewhere like this, if instead it falls out, then it's not falling in the region at all. That's not it. Then if it's falling outside. So basically I've defined this region, you know, region start to region end. This is the region, and we're trying to see, does this SNP in this location, like, where does it fall? You know, we given the location and we also know the region start and end. For the same, let's assume that the chromosome is the same, you know, for the same chromosome. So for the same chromosome, so we're holding the chromosome constant, and then we're trying to see, you know, does this SNP fall into the interval below? That is the question that we're trying to answer. Like, does it fall outside the region? Or it could also fall like outside the end over here too. Or does it fall within, you know, within this region here of interest? This is the region of interest here. You know, we need to see if it falls within this region. And again, in this spot, it falls in the region, but in this spot, it does not fall in the region. And these regions can be used for so many things. These regions can correspond to like introns. Only 2% of the DNA codes a protein. The other 98% does not. It's non-coding, which is sort of really interesting. Like, what does that really do? What is the job of that non-coding DNA? You know, introns, all these, you know, retrotransposons, simple sequence repeats, segmental duplications, LINES. You know, there's so much that's going on here. And in fact, you know, when you look at a DNA region, you know, there are like regulatory sequences, there's a start and end, there are these introns. Exons are really what goes together to help form the messenger RNA transcript that then is translated by the ribosome into a polypeptide. But you can see there's a promoter, 5' untranslated. These are regulatory sequences along a DNA strand. So we're just trying to figure out, like, is there an overlap? So basically, I have this file here. This file has these SNP IDs. So it's my GenomicRanges file 1. And this is for attention deficit hyperactivity disorder. It's a genome-wide association study. So attention deficit hyperactivity disorder, which is basically, it's just basically associated with, you know, being easily distracted, appearing withdrawn, forgetful, exaggerated emotional responses, hypertalkative. You know, you have like a lot of symptoms. It's attention and hyperactivity disorder as well. So, you know, that's ADHD. So, you know, you can daydream, you have trouble sitting still, you're forgetful, careless, you can be risky, may have trouble getting along, being disorganized, talk a lot of symptoms here of ADHD as well. And people are trying to research, okay, well, how does it affect the brain in different ways? So this is a genome-wide association study for ADHD. And I have a set of chromosomes. So chromosome information, 1, 2, 3, all of them, you know, till 22. I don't have the gender chromosome, the X or Y, I don't have that. But for each SNP, as I said, it's just width is 1 because it's one DNA base pair and it's given some ID. So this RS11264243 is at chromosome 1 position at this position on chromosome 1. And if you see the carrier type, you can also see that all of these, you know, you can see that these chromosomes are basically like having different, you know, 1, 2, 3, 4, 5, you know, this is like along a chromosome and there's millions of base pairs here. So the SNP could just be a very, very tiny one base pair difference and we have given them IDs. You can see that all the chromosomes here, you know, we are given 23 pairs. This last one, if it's XX, you're a girl. Otherwise, if it's XY, you are a boy. So, you know, we're given one from each parent. So basically, this is the SNP ID, this is the source, right? This is, and the width is 1. Because again, you know, the single nucleotide polymorphism is just, it's given an RS number, it's 1 base pair only. So we have that file, that's my file 1. You know, the SNP information, SNP IDs, the SNP IDs, the corresponding. So usually this part here, you can usually get one from the other. So this part, you can always find this position and information. You can always find this information from the SNP ID. You know, you can, and vice versa, you can map between the two. You can always map between the two. So you know, SNP ID is going to tell you the chromosome and position, and you can also use these to infer the SNP ID as well. And that's just a unique ID. But you have to make sure you have the correct build. So I have to make sure that these are HG38 as well, that the builds are the same, the mapping, the coordinate system used should be the same. So this file has the SNPs, and the next file has a set of regions. Like, as I mentioned, that there's so many different regions that we can have, you know, in the DNA. But I'm just using some code for some different like made up regions that I created. So, you know, transposons, retro, you know, like whatever regions there are along the DNA, introns, protein coding genes. So these are just some, you know, fake regions. I just made up these promoter enhancer, intron, 3' UTR. I just made these up. So this is my file 2. So file 2 can just have a variety of different regions, DNA regions. So this is just made up. And I have a start position and a stop position and a chromosome number. So what I want to do essentially is determine, can I find genomic ranges file 1? So each of these is with 1. Can I somehow find if this part here, I want to see if I can map. Maybe I'll stick to this. I'm trying to see, does this fall in the SNP? Does it fall into any of these, intervals? I'm looking along, you know, chromosome 1, and I'm trying to see, does it fall within any of these intervals? Yes or no? That's what I'm trying to do. And I have so many SNPs. So my task is essentially this task here. It's just taking each SNP, and it's from file 1. And it's trying to see if it falls within anything from file 2. So that's my goal, having this set of SNP positions and seeing, do they fall into my set of intervals? So this interval is 293 base pairs, 315, 405, 209, 631. These are my intervals, start and stop regions. So these are corresponding to region start, region end. You know, start is the start and stop is the end. And I want to see how to do that. Well, what I can do is I can use this package, and there are many ways you can go about doing this, but I can use this package called GenomicRanges package. You know, it represents genomic ranges in bioconductor. So if you don't have bioconductor, you can install it as well. And you can install this package, GenomicRanges. And it's going to show you how you can represent chromosomes as SEQ names, ranges, strands. And there's all of this, but I kind of want to walk you through the process of doing this. So how do we go about doing this? So again, as I was saying that, you know, so this is my code for overlapping two regions of genetic information using GRanges. So two regions, you know, of genetic information, the SNP file, which is file 1, and file 2, which is the regions, you know, for those, you know, what part of the DNA, and that was made up information. So if you don't have the package, you can just write BioC Manager install. So this is using RStudio. So I have a video up about how to download RStudio. So please check that out if you have any questions on downloading RStudio. So then you can input this information, file path 1, file path 2. So what I have here is I have a variable where you can put in the file information for file path 1, like where is that on your computer, and file path 2, where is file 2 on your computer. And if you have any questions on how to find that, Then what you can do is you can just see like that the file location, it typically comes where, I can click the properties for, like, if I'm going to Genomic Ranges File 1 properties, I can click properties and I can see the file path to use. It's on my C drive, users, Saniya documents. It's cutting off the A. I have this here, and that's how I kind of construct this part here. I can just construct, this is my C drive users. I use it double / on a Windows, Saniya documents genomic ranges file 1, and it's the .csv. And I do the same thing for file path 2. So then for file 1, sorry, this should be 2. And 2, sorry, 2 and 2. So then for file 1, the information that I need is I need to know from file 1, what is the name of, and this is just how I want to construct it and make it a little bit more organized for you guys too. So what I need to know is, what is the name of the first column in this? The reason is when I will read this file in and I'll show you, sometimes the first column gets a bit distorted. It can look like I ... SNP ID. Sometimes that happens. So if you could just note this down, and I want to do this so that you guys can just run this and not have to worry much. So if you just note down, what is the first column name in file 1? It's SNP ID here in Genomic Ranges file 1. For file 2, the first column is instead chromosome, but short by CHROM, chrome. This is chrome, start and stop, so it's chrome. So that's why for file 2, I'm just going to put CHROM because that's the first. So basically, all I'm saying is that this is mapping here, the chrome, and this thing here is mapping to this, the first column file name. So then what I next need to see is, okay, I have that information. So let me just continue on with file 1. So now I'm just going to keep track of, what we've done so far. So we talked about this part here. We talked about the first column there. Great. So now what we're going to see is that what is the Chrome call name? So the 3 columns that we're going to need for the overlap, we're going to need these 3 columns from file 1. And then what we're going to essentially need from file 2 is also the same. We're just going to need chromosome, the start name, and from the width, because really what the width is the width is just the difference between, the width is just the same thing as, stop minus start. You see a 293, it's just the stop minus the start. So it's just the stop minus the start. That's what the width is. So if you know the width, you can just figure out, like the code can figure out the end position, the stop position automatically. So you just need these 3 from both files. For the overlap, right, you just need to know, I mean, it's implied that it's 1, but you don't just necessarily have to be overlapping a SNP. You could have another file of another set of regions. In file 1 could have like a start and a stop position as well, and a width. So that's why it's important to just keep the practice. Like if you might be intersecting 2 regions as well, complete regions. So you might need to have the width as well. So it's good to just have the practice of having a width column, even though all these widths are 1. All of them are 1 for the SNP file. So we just need to figure out the column heading names, just because you might call yours differently. So that's why for Chrome call name in file 1, I put chromosome because I've named it chromosome in my file 1. For the start column name, file 1, this one, I call it position because I've named this file position. This column position. This position is, so basically chromosome is the name of the column with chromosome information in file 1. Position is the name of the column with the position information in file 1. It's just the position. It could also be the start. I can also call it with the start position. And because it's a SNP file, it's the same thing. Okay, great. So that's what position is, just a starting position. I call it position. You can call it whatever. If you call this position, start, for instance, then you can just, change this to being position start, like that. If you've named it like that, position start, then you can do that. We're just going to go back to the way it was. So that's position 1. And then you have a with column. So the name of the column with information. Sorry, this should be with information in file 1. And I call it with. That's great. So we finished with file 1, kind of getting the heading names. And this little guy wanted to learn with us too. Hi, Auntie. Oh, he's saying hi. My little guy. He's very, very adorable. Okay, so that's a little treat for me finishing with file 1. So the next is file 2. So similarly, what we're going to see in file 2 is we're just going to get the names of the column with the, and so this is file 2. So the name of the column with chromosome information in file 2, that's called CHROM, which happens to be the first column. The name of the column with the start position in file 2. So what that is it's just called start. That has a start position. So I just call it start here. So you can notice that this is chromosome, this is chrome, this is position, this is start here. So it may just differ just based on how you name them. You can name them the same way as well. It doesn't matter. So this is start here, and with is the name of the column with information in file 2. So again, that's just how we get each of these. This chrome is here. And yeah, just in case you just wanted to see that again, this is start, and this is with the column names. Okay, great. So now we have the set of column names. So I'm just going to run this information right now. So I'm going to load library genomic ranges. And then I have file path 1, file path 2, first column name. So you can see how this is being filled out with the values of these so far. With. And then what I'm going to do now, so I've written out this code, so that's all you'd ever need to run. Once you have, if you write the code how I'm showing you, that's all you'd ever need to do. And then you can run the code below and you'll find the overlaps right away. So then you just read in df file 1. This is 5,046 observations of 6 variables. So if we just click this view button here, so you can see what I was showing you, that it sort of has this funny number in front. That's why I took down the first column name, just because when you read it in R, sometimes it reads it a bit funny. But it's just as we have it here, 5,046 entries. Yep, 5,046 entries, not including the title name. And you can see that that is file 1. Great. So, and then we can also read in file 2. Oh, I have to. Okay. Yeah. I just named it file 2 like this by accident. Okay. So I'm just going to read this in now. Okay, great. Now I have file 2 and it's 49,000, so many observations here. And you see that this Chrome also gets a little messed up with this II. I'll also show you, that's why we wanted that, right? So if we do the head of file 1, and this is also another way, guys, like if you just want to use the start and stop columns, this absolute value would help you like similarly figure out the width. You can always do that if you need to find out that information. And this is the head of file 1, and you can see it has this weird I in front of it. So you can see if I do column names, it'll give me the name of the columns. And you can see that this is not quite SNP ID. It's not quite SNP ID. Something weird happened, though the other names got preserved: SNPs info, chromosome position, width, file num. I had never figured out why it does that, but if now we can just change it to what we wanted it to be, which is SNP ID. So we're just reassigning it here. So that's what we did here. We're just reassigning it. So please fix the first column name for file 1 because it may be messed up. That's what this says. So I'm fixing it. So now when I call this again, it hopefully is SNP ID. And head is going to again show me the first 5 rows, and we see that we fixed the first column name. So I just did that because, like, you know, when you're doing the overlap as well, like this one, you know, for instance, for file 2, this Chrome would have really been messed up because it's not like, it's different from this column name is different from Chrome. So it's just good to just, you know, note down the column names, and that way when you load it in, you can just make that adjustment. So we can view the column names here. And if we view this as well, we'll see that it's like some funky dot Chrome. And the first column that we wanted to call it was Chrome. So we had noted it down before. So now we can just see the column names again. It's updated. And now if we do the head, we can see chromosome start, stop with region annotation file num. So you can do Control Enter or Command Enter to run code, or you can just hit this run and it will run. You can highlight it and run it. If you want to, you know, directly run multiple lines, you can just do that. Okay, great. So basically we have noted down the file paths to load it in. We have also, you know, found file 2 information and file 1 information on the heading names, and we just read in the files and we made the adjustments for the first column because they were looking weird. We just made those changes. And so now if I look here again, you know, I can just see that this is oh wow, it automatically got updated, right? That's great. SNP ID and Chrome. And you can also just like sew it on things that you want to. Okay, great. So now we are trying to define the genomic range for file 1. So we're trying to make it like a genomic ranges file object, which is what we have in file 1. You know, like we're trying to make it into a GRanges object, like something like this, for instance. So what I want to do is I'm going to use this thing that's called grep. Grep, what it does is it searches for the presence of something in a list or a vector. So if I have a set of column names for file 1, what grep is going to do is it's going to look up chromosome in this vector. So basically for grep, you have the term to look up, and you have vector with terms. And grep, what it will do is it's going to return the position or positions of the term. Now, if you want to look up an exact fit for something, like let's say I want to look up, I could also just do grep. I could do NP. Oh, 1 second. And I'm going to get this 2 over here, NP. I can also look up like ID. And if I look up ID and call names of this, for instance, Y is going to give me 1 and 5 because what happens is that it's going to look for the word ID anywhere within this vector. So it sees, hey, there's ID over here in this first element, and there's also ID in width. You know, there's a term ID over there. So the way that sometimes you can avoid that is, so you see it returns both of them. But if we're just looking up NP, then it's going to return 2 because 2 has lowercase NP. That's why this returned just a 2, because it can be case sensitive. If you want to do exact searches in using grep, you can always attach this special thing in and outside of this. So if I want to just do like this, it's just going to give me, so you have to have this //b here and //b if you want it to be exact. Like if I'm looking up SNP, and like let's say that this was SNP's info and then this was SNP underscore ID, and I just, or this could be SNP and then this could be SNP's in cap, like let's say that instead, for instance, like let's say that we had SNP, and then we also had SNP info, and then we had other info. Like let's say we had this fake vector, random vec is equal to this, okay? And let's say I just want to look up, I want to grep I can grab SNP info in random vector. And what it will do is it's going to tell me 2 because this is SNP info here. But let's say I want to just get the SNP column, then how would we get it? Like if I look up this, I'm going to get this and this, and that's not what I want. I just want the column that has SNP in it, not both of these, because these both have SNP capital in it, and this one. So what I would do is I can just do a dash dash B and dash dash B, and I can get 1. So it's an exact thing. So you typically add dash dash B here and dash dash B here for an exact search. So you just need to add in those dashes if you want to have an exact search. So that's all that grep is doing. For us, it's trying to look for so basically that's why I want to do this, so that, for whatever column names, because this may or may not be the first time you guys, or first or last time you guys will ever need to do this. You may have to overlap not just these sequences here, like these 2 files, you may have to do this for other files. So maybe in one file, you don't remember what you called it. You called it position here. One time when you're doing this, and next time you call this, maybe you're doing it like you're calling this like lowercase pause, and you don't want to have to keep changing the code too much because you changed it. You just kind of want to mention it above that, hey, yeah, I, just make note that, for this file, the only things you'll have to change is this. That's what I'm trying to ensure with the code. The only parts you have to change are just these parts, the file paths, the first column information for each one, and just what each of those 3 headings are called. The chromosome column, what is it called? The position column, the width column for file 1, and the same for file 2. Because we're going to use grep. We're going to use grep. And again, guys, just showing you that this //b is how you do identical searches. And you can see that when you do that in the random vector, which has all of these, you get only the one that you want back, which is SNP. So now what we can do is we're just looking in column names of this one. We're looking for chromosome call name file 1, which is what we specified above, that it's chromosome. So we're just looking here to get the column number. So this is the column number for the chromosome number in file 1. That's what I have noted this as. So when we do this grep, this is going to tell us, hey, the 3rd element in this column names, the 3rd, this is index 1, which is different from Python that starts at 2. That starts at 0. This is 1, 2, and 3. The 3rd column is chromosome number. It's a chromosome column. We were looking for what you guys put in, but you put in what the name is of the column, and it was hunting for it. And then for the chromosome vector value file, all I'm doing is I'm going into, so basically in a data frame, you have rows and then columns for it. So here we're taking all the rows because there's nothing before the comma. That means that all the rows are selected. If you had to select certain rows, you could do like first 3 rows. That's 1 to 3, for instance. So we're taking all the rows, but we're selecting specific columns or column. And the column that we're selecting is column 3. So we're just going here and it's going to give us this set of, chromosome values from DF file 1. If you look here, we're just going and we're getting this value here. And note, we could have easily just done this, guys. If you're wondering, we could have just done this and we could have used these values. Oh, DF file 1. We could have just used this right away. And instead we could have just said, hey, these values are this. The only reason is I just want to, you can do this. That's also fine. You can just do that. But I just want to make it in case you guys change the name of the chromosome from being like, like if your chromosome file, maybe you call it CHR or it's something else, right? You don't want to have to keep going through the code and changing it. So this just gives you the set of values. And the start vectors, again, what it's looking at is the same thing. So you're going to notice that I'm doing the same thing here. This is the same thing. The grep is all the same. It's all the same here. It's just that whatever I'm looking is, whatever I'm looking up just differs, that's all. It's all the same. So for the start, I'm trying to get the start vector values because remember, as I was saying, that I want to get chromosome position and width. I need to get those from file 1. So I'm getting the start vectors. I'm looking up within that. I'm just looking up position, and I'm figuring out, you know, in these column names, I'm getting that position is 1, 2, 3, 4. And it's a way besides hard coding for you to just look this up. And it's going to always be robust. It's always going to be reliable than hard coding in column 4, because we could have just easily put 4 in here and then done that. Or we could have just said that this is the same thing as converting it to numeric. It's the same thing as df file 1. And then it's the same thing as just doing position. I can also do this. This would also work for me. This will give me the start positions. But again, guys, you can do this. You can definitely do this. But I just want to make it more robust for you guys, so it can work. So this is giving us, file column 4, and then this is just extracting that 4th column, and it's giving us those start positions. So you can see like this is this value 154, that is the same thing here. 154, it's the same thing. So, and also for the width, we're just looking up the width column, and we're finding out that it actually is the 5th, because if you look here, it was the last column, it's second to last, 1, 2, 3, 4, 5. The same thing here, you could see it's 1, 2, 3, 4, 5, 5. And that's how we figured we basically have the set of those values here. So we have the chromosome values, we have the star values, and we have the width values. Oh, I have to run this. Now we have the width values too, which is just 1, but again, because it's SNPs. And if you didn't have it, you could have also just done this, width is equal to 1, because it's SNPs anyway, so width is 1, but I wanted to make it robust for you guys. So now we can make a GRanges object for file 1. So this seek names, this is, as you guys may have seen, seek names is just what GRanges uses to call chromosome. So it's really important, guys, that one thing that you do is if this chromosome, they could have called it like CHR1, CHR1, they could have done it that way. In that case, you should make sure though, that if this has CHR1, but this one does not have CHR1, it just has the numbers, that these two should be consistent. You have to make sure that, if this is just using numbers and it doesn't have, sometimes they can have, like in that example, they can have CHR numbers in front. So if that's the case, then you just want to make sure that these two are consistent with each other. Otherwise, you'll get errors if one has CHR in front and other one doesn't. And you can just do like a command find replace, and then you can replace like CHR with just blank space. If in that case, if it can open in Excel, you can just do a quick replace. So that's how we can build out. So seek names is always going to be the chromosome values. That's the chromosome values. The start is just going to have the start values, and the width is just going to have the width values. So when we put this in, this is the GenomicRanges, GRanges. So we look at this file, and what it looks like is it has, all these 5,046 ranges, 0 metadata values. So everything here is important right now. Seek names, the chromosome numbers, the ranges, and the strand. So this is all the information from Genomic Range 1. So we do the same thing for like file 2, which has those regions, the chromosome start and width. So again, we look up, we have the column names for file 2, and we're looking up the chromosome because we mentioned that CHROM was the name, the chrome call name in file 2. We mentioned this above in the user input. We mentioned that there. So that's what we're going to look for here. We're going to look for using grep. It's one of those Linux commands or smart commands to look up something again. So we're going to get, hopefully it should say 1, which it does. And that's what we're going to look at. We're going to take that first row, first column, rows and then columns. We have taken all the rows because there's nothing in front of the comma and just the first column. The vector of chromosome numbers in file 2. And then we also have, so that's what this is. And then we also have the same thing for start column. So we're looking for this start within these column names. And that's going to be the second column, which is what we should get. And again, you can do it. You can always just, you can just always say that, you want this to just be equal to, there's nothing wrong with you doing it this way. If you just want to do this, that's also fine. You can just do start and you can just, it's the same thing. But the thing is that what if you don't call it start and some other time you're running it, it might just crash, and it's no big deal, but I just want to make it so you write it once, take care of the headache right now. You don't have to, worry about having to do this again. The only thing you have to just make sure is that, you can just make adjustments to this above as needed. Each time you run this, just make sure, if there's any changes to those column names, and then you can just like run this, like just, after you finish this, you can just run everything below and then you'll be done, in just one shot. You don't have to rethink any of this again. So you have your start vector as well, which should be the 2 because of the column names, the names of the columns again in this as well. And then you have the width column vector, which is looking up the width column name that we specified, which is also called width within this. And width is going to be 1, 2, 3, 4, the 4th column. And that's what this is. This is the same thing, it's just putting a 4 here because that's what it became. And we have the width. So now we have chromosome values vector, we have the start values and the width values. So we can similarly construct the genomic ranges for file 2 using, chromosome vector values for the seq names. We can use the start values vector for the I ranges. So we're defining an I ranges object that uses start and width. And then from that, it can infer the ending, because the ending is just the same thing as, end equals start plus width. That's how you get the 2. And width, and similarly width, it just equals end minus start. And if you need to put that in absolute value, you can just do absolute value of this, to make it positive if for some reason, the signs become negative. So because sometimes the strands, the direction positive and negative can differ. So this is GR file 2. So we can see that it has, also the gender chromosome Y as well for males. So this is like 49968, right? That's great. So now we have, so basically we have GR file 2 here that we showed, and we also have GR file 1. And now what we're going to do is we're just going to find the overlap between these two. So again, this is in this situation, this is just, DNA regions. And then this other guy below right here is just, this is just SNP positions. And we're just going to see how do these map, do any of these overlap in any of these ranges, controlling for a chromosome and a range. How do these positions, do they fall inside any of these ranges? Where are their overlaps? So then we can figure out where the SNPs, for ADHD, where do they tend to fall? Like which regions are they in? So that's what we're trying to do there. So now we have both of them. So now we're finding the overlaps between both file 1 and file 2 based on the genomic regions. So there's a find overlaps function in the genomic ranges package. And the way that it's set up is query comma subject. And it doesn't really matter which one is which. It's just that like the way that genomic ranges, GRanges deals with it might differ. So, it's not differ, but it's just like it refers to the first thing you put in as the query and the second thing as the subject. So here we put GR file 1 first. So it's going to say that that's the query file. The query file is GR file 1. And the subject is the second argument here, which is GR file 2. So this is the subject over here. So that's what it says. So then we take an overlap between them. And that's what we get. We get an overlap object where it's figured out the query hits and the subject hits. So which rows from the query, and the query again, guys, is the GR file 1. This file 1 is the SNPs file in our situation. The file of these little cute little SNPs here. And the file 2 was the DNA regions that were made up. And then this is just like DNA regions that I've made up. And that's what the overlap is going to tell you, like, that this 16th row of the SNPs file actually matches the 6,768 row of the DNA regions. Like that's what maps to what. That's what it's telling you, that, hey, this row from the SNPs file actually also maps, like it falls within this region of the subject hits. So the other way to see that is like if we have DF file 2, and the row that we were using is this one. And then if we have DF file 1, and what we'd seen is that that this thing matches 16, so that's 16. So what we should observe is that from the DF file, this region, row, again, first thing is row, then columns, that on the same chromosome, so that's great. Chromosome 11, that's great. And this is the start and stop, okay? And and this is the start and stop region, which is about 1,290 base pairs. It's some made up region 34. But this position, it seems like this may actually fall, this is this region here. Actually, this is a case where this position, it falls in the interval 3772633, 772633. It falls within this interval. So that's what we have gotten, which one's mapped to which. So it falls right in the interval. It's great. And that's what that data frame is telling us. So it's telling us, for the ones that fall in, the ones that have fallen in to the interval, like which rows do you combine together? That's all it's telling you. So that's what my overlaps, that's what my overlaps is trying to tell you, which rows do you join with which, And again, you could do the same thing with 19 and this as well to see that, that they do map together as well. So rows to use for file 2 just come from subject hits because GR file 2 is the subject file, again, because of the find overlaps query and subject. And that's what the logic is, what I mean, that you just have to do this once and you don't have to remember the logic, like that again. That's what I meant. That quite can be tricky and you just have to remember which is which. And so that's what you do here. This is the subject. And if you forget, you could just see that the first one is query hits, which also corresponds to what we put first, which is, GR file 1. And the second one is, subject hits, which corresponds to what we put second, which is GR file 2. That's another way to remember it. So based on this, so these are the rows to use from file 2, and you can see that it has this, it has this 6, 7, 6, 8, and then all of these things. That's the rows to use. Those are the rows to use. And rows to use file 1 will come from the query hit. So this should have 16 as number 1 and the other ones. And then for the overlap, all we're doing is we're taking data file 2 and we're taking the rows. So remember we were doing columns before. So now since there's nothing after the comma here, we take all the columns and then we take all the columns from file 1. But we are selecting the files from row 2, from file 2 based on these rows in this order. And then, so that's what this is doing. This is selecting all those files that, like the first one was 6768. Here, this region that we were looking at before, it's selecting the rows in the order given by this vector. So it's going through, and the first element will be this and this and this, which is what you can see here. And you can see that the same region is used twice, here. It's mapping to multiple SNPs. So that's something that you note as well. So that's why, and this one is just taking the rows to use from file 1 and going through file 1 and selecting those rows in that order. So this is 16, 19, 27. They may or may not be in order per se, like this, like in file 2, this is 865, but the first one is 6768. It doesn't matter. It's just the ones that happen to map together. And that's how you can select. So this one is going to give the set of rows from file 2 and the orders in that regions file that happen to map to those respective rows and orderings in file 1. That's what we're getting here. We're just mapping them together using that overlap rows. So this overlap BF will have what I was showing you guys, which is exactly what we want. You know, the 6, 7, 6, 8 here for the, you know, the chromosome number, start, stop with this stuff right here from file 2. And you can C-bind it the other way. You can C-bind it with DF file 1 first, and that you don't have to go this way. So this one is from file 2, and this one is from file 1, and all of these should work. Like we went over this first one, where you can see the SNP. The SNP falls within this interval. So this position here, it falls within this interval here. So that's great. And now let's see another one too. Let's just see what happens here. This region of 2, 485. And this is all on chromosome 2. So the chromosomes must match. So all these chromosomes must match these because it's the position on that same chromosome. So, and that's what it needs them to be in the same format for, you know, if it's CHR1, you know, instead of 1, you know, one file, then you have to make sure it maps in the other. So here, this is 231,611, 231,856. So again, that's the way to see that. The region is 231,611, and then the end is 231,856. And if we have the SNP, so these two other regions here, and if we have our little SNP here, that is at 231,741. It does indeed fall. It does indeed fall in this interval between the two, between these two regions. It does. So that's great. It falls in between them. You know, 611, 856, 741 falls right in between. So you can quickly verify that all of these, essentially all these SNP positions, you know, given by this ID and chromosome and position, they do fall within these intervals. So that's awesome. So this shows that we can find the overlap of it. So we can do a head, and then we can also just see, like, again, that this is the same information. This is the information up to file 2, and this is the information up till 5.1, and they all are mapped. And we have the dimensions of the 86 rows that we found. So please note that we found 86 regions shared by file 1 and 2. So we can also figure out maybe how many SNPs mapped. So we can do length, unique, overlapped, DF, and then we can just do the SNP ID, and we can figure out, well, okay, there are 86 total rows here. So like SNP and region combinations, but does correspond actually to 46 unique SNPs, 46 unique SNP IDs. So those SNPs might be found in multiple regions that I created as well. So that's just an example, and you can just write out this overlapped DF to some file here. You can just write it out, and you have your overlapped DF. You can view it as well. You can write it on a CSV, you can view it, work with it, and you can also just like see that, yep, they do fall within the interval. So yep, we essentially found the SNPs that fall within certain intervals of a region start, end, and end. So again, guys, that's just how you find the overlaps between you know, two regions, and they don't just have to be a SNP in another region, they can be anything. But I just hope that this was helpful for you guys. So this is the GenomicRanges GRanges package. So I'm just going to quickly show you again the code. Just so if it is helpful for you guys just to see it again, I'm just going to scroll through it. So, you know, you can either install or have the library, have your, you just need this information here, and then you can just run everything below if you set up the code properly, like how I've shown you. The file paths loaded in, the information on the first column names, and where the start and width is. You just need the chromosome number for this purpose. You know, if you're doing what I'm showing, like finding if it's within an interval, chromosome number, start, and width. That's all you need, those three informations. Pieces of information. You just need the chromosome position and width from this one file, and then chromosome, and then the start and width. That's all you need is just a position, a width, and a chromosome number, and it's just going to overlay the regions. And then you just need to read in the file paths. It'll do that. It's adjusting the first column names for both files. And then it's just looking up the exact column numbers where all those chromosome numbers, start, and width are, and it's constructing a GRanges object using that, where seq names is a vector of chromosome values. This ranges is an iRanges object that has the start values and width values. And you have GR file 1, and then you also have GR file 2. That's just the same thing, but just for file 2. Then you take an overlap of GR file 1 and GR file 2, and you can switch them up, but you have to remember that find overlaps is doing query comma subject. So you find the overlaps between them, and they're both GR objects. So this is good. You can do it this way. And then you just have to figure out the rows to use from each file using this. And then you just map them to each other, and you C-bind it, you column bind it, you smush them together. And now you have the overlap data frame. So those are just the steps again that you do. And I hope that this was helpful. And please let me know if you have any questions on this. So my name is Saniya, and I hope that this was helpful for you guys. Thank you, and have a great day. And also, guys, there's some additional documentation and additional functions for more complexity using genomic ranges as well. Exons, binding sites, peaks, reads aligned without gaps. There's so much here that you can use. And there's a lot of functions, introns, gene models, unspliced transcripts, gene regions, disjoint bins, promoters, introns. So there's more complicated things you can do. But I just hope you can understand the basics of how you overlap two regions, especially like a snippet or like one position base pair inside another, or see if one is found within the other, for instance. If you have like maybe a short sequence, see if it's found within larger sequences, for instance. So I think in those situations, the query and the subject could matter depending on which one is longer and which one you're trying to see falls within the other. But Genomic Ranges is going to try its best to map them to each other. So I just hope that this is helpful for you guys, but there's definitely like so many more things that you can do with this that, once you have the basics down, hopefully it's just going to be just a matter of applying it. And I'll try to see if I can create more videos on GRanges. I had to do it on some work I was working on as well, but and it was sort of a bit confusing at first, like what's the subject, what's the query, like how do you construct it? But when you get your handle over it, then I'm sure it'll be much better for you guys, and it's super efficient. Because I know me, I was trying to write Python code, trying to avoid using genomic ranges. It was very confusing, but once you just deal with it, it's not going to be as bad. Touch wood. So hopefully it helps you guys. And there's just so much out there that you guys can see, and you can see all of this stuff. Wow.